So population genetics is covered in chapter 23 of the Campbell textbook if you want to read about it. But basically it's going to be based on math and using math to give us a way to test whether a population is evolving or not, right? So I know I kind of mixed the lectures up, but I kind of like doing the lecture yesterday, the speciation lecture, the last 2 days. And I might keep it that way actually, and then do population genetics after that one, and then do speciation 2, which is the next lecture. Because what I've kind of spent some time doing is talking about how populations evolve, both microevolution and macroevolution, and then how we get speciation to occur. And a lot of the terms in this lecture are basically already been defined in the the last 2 lectures, basically. And we'll combine some of our already understanding and using some terms that we used in meiosis and some terms we used in genetics. And so we're interested in looking at how we can test that evolution is actually happening. And to do that, we kind of got to make sure we got all of our terminology down, right? Which we've already kind of went through, but it's nice to have slides for it. So 2 types of evolution that we've kind of talked about is microevolution versus macroevolution, correct? So microevolution is kind of where we focused a lot of our time outside of the last lecture, which is within a species, which a species typically exists in populations, which we're going to define in the next slide. Within a species, allele frequencies change from generation to generation. And we have tons of evidence for this. You just need to look at a population and you can see that phenotypes change from one generation to the next in species that we can observe. And we know that genotypes control phenotypes typically, right? Not all the time. I gave you a few examples where that's not the case. But if we assume most of the time genotypes are what are causing phenotypic frequencies within a population, then we can basically see that from generation to generation. Now, macroevolution happens more slowly. Microevolution happens quickly. We can see it quickly, correct? Macroevolution happens much more slowly and happens above the species level. So remember taxonomy: kingdom, phylum, class, family, genus, species. Anything above species, that is looking at 2 or more species or above, is called macroevolution. And in order to get 2 species from 1, we talked a lot about that in the last lecture, we have to build prezygotic and postzygotic barriers that prevent breeding, right, between 2 species to treat them as individual species. And to do that, we have to get a bunch of accumulations of microevolution. So basically, these little events that change allele frequencies can eventually accumulate over a long period of time to cause macroevolution above the species level because it produces those pre- and post-zygotic barriers that we've talked about. And so when you look at, for example, finches, these are all of the Darwin finches here, or a number of them that are represented. You can see we have some insectivores up here. We have some seed grinders down here. Here's the big dude, the large ground finch, which basically can eat any really large seed or fruit that none of the other finches can take advantage of. And if that bird can do that, then it's got a food niche that's it's monopolized, right? That it's only manipulated or used by the large ground finch. Because of that, they'll reach adult size and pass that phenotype, controlled by a genotype, on to the next generation. That's kind of what we call directional evolution, right? Or what we call natural selection at the microevolution level, at least if we're talking about a single species. So we want to check whether a population, or have a way to test and gather some data on whether or not a population is actually changing from generation to generation. So to do that, let's make sure we got our terms down. So I keep saying species, population, yes, and so forth. Yep. Microevolution is just a change in alleles and phenotypes from one generation to the next. If that's directional, and you have many of those micro events happening within a species, you could separate species by some barrier, and the accumulation of microevolution in one little population versus another means they can't come back and breed again, and you get two species. That's macroevolution. So macroevolution takes longer and is typically thought to be accumulation of microevolutionary events between populations that lead to new species.
So if you want to put in like a straight bead bracelet and then like micros a little beads kind of lead up to the bracelet, like an analogy form.
If the beads represent different populations, yeah, you have to separate them, and then when you try to bring them back, they can't form one bead, right? Yeah, they can't breed. Exactly. Good? Okay. So what is a population? A population is a group of organisms that are a single species, one species, that can interbreed and lives in the same place, right? So that's what a population is. Individual species, a bunch of them living together, and they can interbreed. So a species can have multiple populations that are separated by mountain ranges, for example, right? Or hills, or roads, or there might be a forest clump here that has a population, and a forest clump here that has a population of the same species, separated by farm fields, for example, right? Does that make sense? A community differs from a population in that it's all of the populations of all of the different organisms within a specific geographic range. And so I've given you a picture here of a typical field, right? That you might see driving down the road with all the wildflowers in it, right? Let's say each one of those wildflowers is a different species, or maybe there are multiple species of wildflowers there. So you have a population of each of those living in the same place. This is a grassland community with many populations of plants and then many populations of other organisms like butterflies, right? Which you see on top here. So this population of butterflies that represents one species obviously has its own gene pool, right? It has its own genetics, and those genetics, that gene pool leads to phenotypes that you see. And in this example, in this population that lives in this field, this species of butterfly has a black phenotype, a white phenotype, and an orange phenotype. You see that, right? Those are what you see when you're making observations. And if we assume or consider color in this example, to keep it simplified, controlled by one gene locus, then we can figure out kind of what the gene pool is if we can test for genetics. Okay? So a gene pool is all of the alleles for a single locus within a population. All right, so remember, a gene locus controls one gene, and that gene controls some phenotype. Here it's color, right? Here it's color. All alleles within a population is the gene pool. Or you can say all genotypes within a population is the gene pool, because alleles make up those genotypes, right? Everybody remember what a genotype is? What's a genotype? And we'll just assume everything's diploid, because we're only going to deal with diploid for now. It's what makes up your genotype, right? And so if we think of one gene locus, a genotype will have two copies, one from mom, one from dad. And if those copies are different, then they're called alleles, right? Or they're called alleles, period. The instructions are called alleles, right? And they might vary a little bit. One might be dominant, one might be recessive, one might show codominance, right? Depending on what alleles you have, okay? So a genotype has two copies of alleles, right? Is that correct? For a gene locus. Just transferring what we did with genetics over to this. Does that make sense? Now, we know, did I cover everything I wanted there? Yes. All right. Oh, selection happens to populations. That's the smallest unit that can evolve, is a populations. Individuals don't evolve, populations do. So you don't evolve, humans evolve, right? Probably not anymore at least, right? We're too selective and too controlling. But some argue we still do. Okay, so we know if you look at a population of ladybugs and you were to pull them out and find all of the variation you see, you might see something like this, right? So there is lots of phenotypic variation typically within a population, which remember is a single species that can interbreed. We know that most, not all, but most phenotypic variation is controlled by your genes, right? What alleles you have. So here are some ladybugs, and this is just a small sample of what our ladybug can look like. Can have absolutely no spots till, almost completely black, really, to be honest with you. But here we got lots of big, large spots. So there's a lot of variation here, which means if we have phenotypic variation, then we most likely have genotypic variation, right? The genotypes must vary to control the different types of patterns and colors that we see. Now, we, as humans, have taken wild grasses, or types of wild grasses, and selectively bred different variation that we see in that wild grass group to get the food items that we like to eat, right? So we did that. We took this wild grass, saw some difference in variation, like leafiness versus difference in flowers, and continued to breed those individuals to get the phenotype we want, right? And then that's what we eat. So cauliflower, broccoli, kale, Brussels sprouts, cabbage, they all come from a single group of grasses, wild grasses, that we have bred over hundreds of years. That's called artificial selection, right? Natural selection can happen to a population of, say, ladybugs if there's a selection pressure that favors one or more of those phenotypes, right? So let's say for some reason these two on the end have a higher survival rate than these three. Over time, these phenotypes, and thus their genotypes, are going to become more common than the other ones, right, in a population. That is what we consider evolution or natural selection at the micro level, right? Microevolution. Now, for evolution to happen, I've told you over and over and over again that selection loves genotypic variation, right? Or gene variation, genetic variation. So how do we produce or maintain genetic variation, which leads to phenotypic variation? There are four major ways, at least your book, the book or how we usually teach it here. There are four major ways to get genetic variation. So, the big question is, well, how do new alleles just pop up, right? I mean, how do they pop up? New alleles form with variations in the instructions. That's all they are, right? So how do we change instructions? We change wording, don't we? If the real instructions. That's exactly what we do here. We change the DNA base sequence by replacing one base with another one. It's called mutation. Mutation happens at different rates in different organisms and different environments, depending on what you're exposed to. We, eukaryotes, mutate very slowly as far as our genome goes because we have great proofreading machinery. So when does mutation happen? It happens during replication or maybe during mitosis or meiosis, right? When you're moving chromosome and chromatin around or copying it. Our proteins are really good at going, oops, I messed up, and then they fix it. All right? Some organisms don't have good proofreading machinery. Viruses, bacteria, for example, their machinery is not as poorer than ours, or it's just not as in tuned to their genetic code as ours is. And they also reproduce much faster than we do, right? So mutations can build up in organisms that don't have good proofreading mechanisms. We could also alter gene number. All right, that is, duplicate genes on accident. If you think about that for a minute, if I duplicate a gene, it's not necessarily bad, right? You just have an extra copy of a gene in a chromosome, maybe made your DNA a little longer, but it really doesn't do much to chromosomes. So altering gene number happens a lot. That is, there's a lot of mistakes that are happen where the enzymes that copy DNA copy an extra copy on accident. It's worse when you delete things, right? When you remove, genes that code for something. So a lot of times altering the gene number is a good thing. So if you need a whole bunch of something, it's better to have more than one copy, right? You can copy more messenger RNAs more effectively if you have multiple copies. You can also alter the position on a chromosome if you have mitotic error and meiotic error that is not too major as far as rearranging genetic information. And that has happened, that it can cause some genetic variation. Rapid reproduction, particularly with poor proofreading, like viruses, particularly things like HIV, which are RNA viruses that can reproduce really quickly and also have terrible proofreading mechanisms, so they mutate really quickly. And then obviously the big one is sexual reproduction. And why most organisms practice sexual reproduction is it mixes up the alleles, right? A little bit. It also enhances these. So if you sexually reproduce and you do it rapidly and you mutate, you can mix those mutations up quickly into a population, right? Because you're passing them on to the people you breed with. So let's take each of these. I have a slide for each of these 4 sources. So mutation is the way we get new alleles. Mutation is pretty random, and it's a random change in nucleotide sequence that escapes our proofreading. Even though we mutate slowly as eukaryotes, we do mutate. Our DNA is mutated. If you give a long enough period of time, like hundreds of thousands or millions of years, or think of thousands and thousands of generations, eventually a mistake is made that's not caught, right? So there's mutation in almost all nucleotide sequences within every organism if the organism has been around for long enough. Now, mutations can be significant in the sense that they alter the instructions enough that your protein does not function properly, right? Or maybe it functions more effectively. It could be a major advantage or disadvantage if you change the instructions too much, right? So that the protein doesn't work right. Or in some instances, it works better. So you can have a significant change that causes something like sickle cell anemia in certain situations when you get one copy of altered instructions. This is for hemoglobin, if you remember. And if you remember, I told you it is what is considered a point mutation. So see, point mutation is when you change one base. Point mutations are very common because you're only messing with one base, right? It's not like you're changing 250 base pairs, you're changing one. And so it's much more likely that proofreading will miss one base over 50 bases, right? If you think about it. And so point mutations happen often. Most of the time when a point mutation happens, it's neutral. It's a neutral mutation. And what that means is it does nothing. It doesn't change the code enough to do anything to the protein. Or it happens in a part of DNA that doesn't code for anything. So you have lots of neutral mutations all over our genomes that really do nothing to our normal everyday existence because it hasn't changed the instructions very much. But every once in a while, you'll get a significant point mutation that does something like sickle cell anemia, where it really alters the phenotype of whatever characteristic or trait we're looking at. So in this instance, remember, if you get one copy of the sickle cell allele, then it replaces one base from the wild type, which changes an amino acid out. So if you want to know, you replace a glutamate with a valine. Glutamate is water soluble, valine is not. And so it makes the protein a little weaker than normal, so that if you get low oxygen, the protein changes shape and your cells sickle like this, which is bad, right? It's called a sickle cell anemia event. But changing out one base for one and then changing out one amino acid for another makes you resistant to malaria. So it has a disadvantage, but an advantage in that you can survive malaria, much higher probability of survival with the sickle cell allele. Now, mutations that are harmful, like sickle cell anemia, often hide because they're recessive. So remember, if you're recessive, you can hide or be masked by the dominant trait, and you might not even see it in the population, but every once in a while it'll just pop up, right? Because it's being passed around in the population, it's just not showing up in the phenotype. And so that's how harmful mutations often hide within a population or a genetic pool, is because they're typically masked. The other thing I like to point out immediately is just because a trait is dominant and just because a trait is recessive doesn't mean one is more common than the other in a population. A population can be recessive dominant because the recessive trait is selected for, right? So it all depends on what the selection pressure is, right? So it's not dominant and recessive as being popular or not popular. It's just dominant when present will mask the recessive, right? So in this instance, what you're going to learn when we get to our next lecture is that the heterozygote, having at least one copy of the recessive and one copy of the dominant, is the advantage, is advantageous. All right? And having that genotype is what makes you less susceptible to malaria. Altering gene number. The second way we can increase or change genetic variation, by simply duplicating genes. This happens a lot with meiotic errors that aren't too major, or just enzyme mistakes that happen during copying. Instead of copying one, you copy two, 3, 4 copies of your gene at a certain locus. And so that you end up with lots of copies of a single gene that has the same function, at least at that point. But having more copies of a gene increases the probability of mutation, right? Because if you go from 1 copy to 2 copy to 10 copies, there's more probability that you're going to get a mutation that sticks in one of those copies versus just one, right? And when they do get mutated, sometimes that means the protein can take on a new function. So let me give you an example of that. So snake venom is imagine I pick a snake, right? Being a herpetologist. Snake venom is or are controlled, the venoms are proteins, and proteins are controlled by genes, correct? The original function of those ancestral genes were to help with the function of saliva. So you may or may not know, saliva is not only wet and watery and produces a salty solution to dissolve what you eat, but it has proteins in it, right? And those proteins have functions. So the salivary proteins are involved in digestion. They help to start break down food materials you eat. Also, a lot of the proteins in saliva are protective. They protect you from bacteria and virus infections. Think about it. Your mouth is warm, moist, with a lot of nutrients in it. It's a great place to grow if you're a bacterium, right? And so you produce a lot of antibacterial proteins, for example, that are part of the saliva that keep bacteria growth down. Now, it would make sense that if I duplicate that, that's a good thing, right? I can produce more proteins faster. So you actually have hundreds of copies of salivary protein genes in your genome. Over human evolution, this gene's been duplicated many, many times and actually selected for because it makes saliva more effective as a solution within your body. Now, snakes have several hundred copies of the salivary genes. Most of those had the same function as your salivary genes. They control digestion and bacteria growth. But a few of them have mutated over the years to then take on new function, like venoms. These proteins are now toxic to certain individuals. So one of the major rattlesnake venoms that destroys muscle is actually a mutated antibacterial protein that now destroys muscle tissue because it can bind to muscle and break open the membrane. Yep.
So like humans just stop like brushing their teeth and stuff like that with those genes keep becoming more frequent
Or they could be maybe under heavier selection if there's mutation. Not necessarily you'll copy more, not that you can't copy more, right? But they might be copied, and then once a mutation happens, it might be selected for, particularly if you have better bacteria fighting proteins. But again, those mutations also have to happen in your germ cells, right? So that's the other thing that I didn't mention with mutations, is most mutations happen to somatic cells and not necessarily your germ cells, right? So why is that important? Why important in germ cells? Yeah, because they can be. Well, if it's in a somatic cell, it won't end up in your kid. So we have lots of mutations in our somatic cells that we don't pass on to germ cells. Now you have mutations in your germ cells. Not, it's mostly male. It's not as much female because we produce so many sperm. You produced a lot of eggs, and if mutations happen in those eggs during development, then those could be passed on.
How do I pass down my mutations?
They'd have to be in the egg cells in order for you to pass them on, or in the germ cells that are egg cells. If it happens in a skin cell, you're not going to pass it, right? So just because I get skin cancer, because I had mutations in my skin, doesn't necessarily mean I'm going to pass that probability of skin cancer onto my kids, because it happened here, right, in my skin and not in my germ cells. Does that make sense? Okay, so another really good example of a gene that's been duplicated that's very advantageous, has been selected for, is odorant detecting genes. So odorant detecting genes are responsible for producing proteins in the nose that are able you to smell or detect odors. We have about 350 copies or so during our human evolution. Rodents have about 1,000 copies. Dogs have more than 1,300 to 1,500 copies. That's why dogs smell so much better than we do, because they have more protein in their nose and therefore can detect odorants at much lower concentrations. And of course, when that happened, it was selected for, right? Because it allows them to smell better. And so again, when you alter gene number or when you get mutations that are advantageous, selection can work on those, right? Rapid reproduction means I can get my mutations out quicker. And if I have rapid reproduction, I typically have poor proofreading mechanisms or less time to proofread. So mutation is more common. And more commonly happens, and not only more commonly happens, it's going to not be fixed by a proofreading system. So viruses and prokaryotes, like bacteria, have much higher mutation rates than, say, a eukaryotic cell, which reproduces, divides slower, and has better proofreading. Because of the rapid reproduction, any mutation accumulates much more quickly within a population of bacteria and viruses, which have short generation times. This is why antibiotic resistance rises in bacteria very quickly, or why a vaccine might not work as well as it should, which is something the general public didn't understand with COVID-19, right? How we kept changing, not only what to do with COVID, but also the COVID vaccine changes from year to year. The general public thinks we just don't know what we're doing. But in reality, the virus changes all the time, right? Because of mutation, which makes it harder to kill or makes it more resistant to previous vaccines. And then the last one, and the most beneficial one, is sexual reproduction, which can help enhance all of the ones we've talked about previously, because sexual reproduction allows you to take alleles from two individuals in a population and mix them up, right? So if you treat a gene pool like a bin with a bunch of alleles in it, if you're mixing those up randomly, or individuals are reproducing randomly, and you're just plucking out, right, different combinations of alleles, it's going to mix up your gene pool really nicely and give you lots of genetic variation. So natural selection or evolution loves sex because it really keeps genetic variation high. These are breeding damselflies, so male and female. All right, so we need genetic variation for selection to happen. Now I want to switch gears a little bit and start talking about, well, how do we test whether a population is changing over time, either by one of those mechanisms or by some other mechanism I haven't mentioned, or by natural selection, right? It would be nice to have a model to use to test whether populations are changing over time. And we kind of have everything that we need. Now we just need to build a mathematical model that we can use to figure out whether frequencies of alleles or genotypes are changing from one generation to the next. Because isn't that what we need to do? Is just, if we have this many big R's and this many little r's in a population that we know, if we look at the next generation, then if those R's and those big R's and little r's are the same frequency, then the population hasn't changed, right? If they're different, then they have changed, right? If they're radically different, they've changed a lot. If you do many generations, you might be able to see a directional move to a particular allele frequency. So I want you to remember a few things that we've already covered. Like Darwin suggested, when a species in a population is let go by itself and it has resources to consume, they will reproduce, and typically reproduce in large numbers. So parents are going to produce offspring, and those offsprings can then survive to reproduce, right? So you're going to have many generations of reproduction, and phenotypes, in this instance here, we have one, a blue circle, right? Will be passed from parent to offspring and offspring to parent and so forth. We also just mentioned, and we've said it many times, that individuals within a species or individuals within a population of a species can vary or will vary if they have genetic variation. So here I've added another phenotype, right? Dark circles and light circles. We know traits are heritable, so a parent that's light blue can pass the light blue color onto the offspring, or a parent that's dark blue can pass that color onto an offspring. Or heck, a dark blue might carry a recessive trait for light blue and vice versa, right? And can pass on the other phenotype, or at least part of it at least, if breeding is random. So we know we can get a number of traits. We also know that traits can be enhanced or move directionally if reproduction is more successful in one phenotype versus another. So here's our original population. Got 2 dark blues and 2 light blues. They breed randomly and give you a population that has more dark blues than light blues. That's passed on to the next parent generation, which then breeds. And obviously there's something here within, if this continues, this trend, that eventually this light blue might disappear completely, right? And then pop up every once in a while. And so there's something selecting for the dark blue over the light blue, right? Some kind of reproductive success, whatever that is. This is the basis of selection. So how can we provide data that supports this, right? That's the question. It's great. You think you can make these observations or suggest this happens, but then you need some experimental test that allows you to gather data to show support for this. All right, and that's where math and understanding phenotypes, genotypes, and basic probability come in. So here is a population. Imagine that, I pick frogs. Love frogs, right? So we got some frogs here. This is my theoretical population, and there are 15 frogs. If you don't believe me, you can count. Hopefully I counted them right. And you notice there are 2 phenotypes, right, in our population. We have red frogs and greenish blue frogs. And so you could go out, catch all the frogs, and then count the number of phenotypes, right? It's pretty easy to do. So there is phenotypic frequency here. You can see that I have kind of, if there are 15 frogs, I have 1/5 red frogs, or 3/15. There are 3 out of the 15 frogs are red. And then the rest of them are blue, or 12 out of 15, which is 4/5 of my frogs are green, right? Now we know phenotypes are controlled by genotypes, right? So we know there are genotypes, and if I could go out there and figure out the genotype for a particular locus, which is very possible to do now with PCR, and all you need is a sample. And frogs, it's really easy to do. All you gotta do is take a Q-tip, catch all the frogs, and do a swab on their back, and you'll collect skin cells, and you could figure out genotypes. It's pretty easy to do. Or take a piece of a plant or some hair or whatever it is, and find out genotypes. And if you look at this, I've given some genotypes here. Say I went out and sampled my 15 frogs and got their genotypes. So we can do genotypic frequencies then. So if you count here, there are 8 homozygous dominant frogs. And so 8 out of 15, or 53% of those, are homozygous dominant. For the heterozygotes, I have 4 out of the 15, or 27%. And then homozygous recessive, I have 3 out of 15, or 20%, right? So that's my genotypic frequency. I could do allele frequencies too, right? Which I would count all the big A's, count all the little a's, right? So with alleles, remember there are 2 alleles for each gene locus, and we only care about one gene locus at a time. So for our examples, what we're going to need to do with alleles to figure out allele frequency and get total number of alleles is we got to multiply the number of individuals in our population that we sampled times 2, right? Because there are 2 copies. It's diploid. If it were triploid, we'd do times 3, right? But we're only going to do diploids. So to get the number of alleles in your gene pool of your population of 15 frogs, we'd multiply this by 2. So how many alleles are in our population altogether? If we have 15 frogs. 30, right? We have 30 alleles for color, either red or blue. And these are my 3 possible genotypes, right? Homozygous dominant, homozygous recessive, heterozygous, correct? So now we can get an allele frequency. So the frequency of big A, if you counted all the big A's up, there are 20 of them altogether in this little population. So 20 out of 30, or 67% of my population, or I should say 67% of my allele pool or population is 20. And then the little a's is 10, right? And so what I want you to notice is these 2 percentages equal up to what? 67 plus 33 equals what? 100, or if we put them into ratios, or we put them into decimals, it would be 1, right? Or equal 1, because we can make this 0.67, 0.33, which equals 1, right? So we're going to give letters to these. So the dominant allele, we're going to call it P, right? And the recessive or lowercase letter, we're going to call Q. We're going to give them letters, as you'll see when we get to Hardy-Weinberg. Now, a population doesn't always have to be completely dominant. It can be almost completely recessive. Right? When a population is said to be fixed for an allele at a locus, that means it's homozygous. And so in this instance, this is another population of frogs where they're all homozygous recessive. So this population's fixed for the red color. Its genotype is completely the same. Is there a lot of genetic variation in this population? Absolutely not, right? We don't want this to happen. All right, if this happens in a population, a single selection pressure that removes this frog is probably going to remove all the frogs, right? Because there's no genetic variation to withstand, if you will, and no phenotypic variation to withstand selection pressures. But it happens. It happens. here's a population with genetic variability, right? Good genetic variation. You got big A and little a's, and you got both phenotypic colors. You got some heterozygotes in there, right? So you got good genetic variation for this population of frogs. Okay, so far so good in our understanding of how things work. I like to build a puzzle for you, right? So that we get to the equation at the end. So let's look at another example. So this has been done a lot with flowers and flower color, because flower color is often controlled by one locus, at least the majority of the color, controlled by one locus. So notice here we have two phenotypes again. We have red and white. Even if we have three phenotypes, red, pink, and white, which would be incomplete dominance, we can still explain it with our three different genotypes. But in this instance, we have red and white flowers, and you probably could guess they're probably in a 3-to-1 phenotypic ratio, right, in the wild, because our heterozygote is dominant here, right? It's got the dominant red color. But if you knew the genotypes of all the red flowers and all the white flowers that you sampled, then you could count alleles, right? And to count alleles here, we simply are going to multiply the 64 homozygous dominant by 2 to get 128, right? Because they got 2 big R's. So that gives us all of 128 big R's. Same with our homozygous recessive. We have 4 of these, which with the 2 little r alleles. So we multiply those by 2 to give us 8. And then with our heterozygote, we have 32. So we have 32 big R's and 32 little r's, right? Then we can add those up. So we have 160 big R's and 40 little r's. Or we have 100 individuals and 200 alleles, right? So you always check your math, make sure it equals up to what it's supposed to. Good? So far so good? Pretty easy. So now we can do our allele frequency, right? Of our dominant trait versus, or I should say our dominant allele versus our recessive allele. So if 160 divided by 200 is 0.8, which is P, right? So P equals 0.8. And then 40 divided by 200 is 0.2, which is Q, or the frequency of our little r. These add up to 1, right? 0.8 plus 0.2 equals 1. If those don't add up to 1, then you did something wrong, right? Remember that. I never checked my math when I did this for the first time and made lots of mistakes. The other thing that I messed up, I'm good at messing stuff up if you haven't figured that out yet, is I confused genotype and allele, right? It's pretty easy to do. So, you got to work with what numbers they give you, okay? Good on getting Q's and P's. Now, if I have these numbers, I took this population out of, let's say we were in a wildflower field, and I have red and white flowers only. I sampled them, and I got as big of a sample as I could, right, for my analysis. So I got 200 individuals, and I genotyped all of them, and then I got this value, these two values for my P and Q. I could predict what the ratio of genotypes will be, both homo, hetero, and homozygous, for the next generation. If there's no evolution, they should be the same, right? They should be the same. Does that make sense? Because nothing changed. If these allele frequencies don't change from one generation to the next, then evolution's not happening, because I'm not changing the genetics, right? I'm not changing genotypic or phenotypic variation. So I could predict what the next generation will look like. Say I wanted to predict homozygous dominant individuals. How many homozygous dominant, or what is the probability that a flower I sample out of the next generation is homozygous dominant? And it's pretty easy to do. If I know P, then these would be two events that are related to each other. So because you get an allele from mom and dad, I would simply multiply 0.8, the probability of a big R, times another 0.8, right? Probability of big R. And I would get, you have a 64% chance of getting a big R from that colored flower. Or if you just think of all of these alleles, these 200 alleles, as balls and you throw them into a bin and you go and you pick one out, I have a 64% chance of picking out a big letter, right? In this instance, 2 big letters, not just one, right? Because if I pick one out, that's 0.8, then I got to pick out another one, 0.8, then I multiply those and it gives me a 64% chance of pulling 2 big R's. Does that make sense? That's kind of how we're treating it. We're treating this not as a Punnett square, but as a population now. Does that make sense? And we're pulling out alleles. So because I do P times P, homozygous dominant would be P squared, wouldn't it? 0.8 squared is 0.64. I could do the same thing for homozygous recessive. If it's 0.2, then I simply could do 0.2 times 0.2, which would be 0.04, right? That's called Q squared. That would be homozygous recessive. So P squared is homozygous dominant, Q squared is homozygous recessive. So now we need a variable, right, for our heterozygote. Now our heterozygote looks like this. And the reason for that is, remember, during meiosis, we have tetrads that line up, right? Remember that? And so we could have the possibility of the tetrads lining up like this, right? Or lining up like this, correct? So you got to take into account that there are 2 ways that you can line up for a single locus, right? Based on whether mom and dad and how they line up at the metaphyseal plate. So notice that that's why you got these 2 examples, 0.2 for the little r and 0.8 for the big R, which would be this one, right? Or 0.8 times 0.2. Or if you throw that into an algebraic equation, it looks like this, and you can reduce that to 2pq, okay? If you don't remember how to do that, just trust me, it's how you do it, okay? So that is your variable for figuring out the genotypic probability or frequency in the next generation of your heterozygote, which would be 0.8 times 0.2 times 2, right? Which is 0.32. That equals this equation, which is called the Hardy-Weinberg equilibrium. So P squared equals the frequency of your homozygous dominant. 2pq equals the frequency of your heterozygote. And then Q squared equals the frequency of your homozygous recessive. Those all will equal 1, or 100% if you use percentages, or 1 if you use decimal points. So if I give you a problem and you solve for Q, you can get the rest of these, right? The one thing you want to make sure when you're done and you figured out what P squared, 2pq, and Q squared are, make sure they equal 1, right? If they equal more than 1 or less than 1, you did something wrong, correct? So this mathematical model that was created by Hardy-Weinberg, Hardy and Weinberg. This guy's a mathematician. I don't know, why do mathematicians always look pissed off? You know, this guy looks mad. Always look mad. And this is a physician. Here's a physician. Both these guys were not only interested in math and medicine, but they were also interested in the evolution of alleles, and particularly phenotype versus genotype. And they're the ones that built this mathematical model that provides us the ability to test whether a population from generation to generation is evolving or not. Okay? We just subtract it from 1, right? We subtract this from 1, and that will give us P. So now we'll have Q and P, which are our dominant allele and our recessive allele frequencies, right? Then we can plug those into here, and that'll give us our genotype frequencies for our population, right? So we let that population breed again, let it overwinter. The next year we do the same sample, count the number of white flowers, get our Q and P, plug them into Hardy-Weinberg and see if those numbers differ from the last generation. And if they don't, then the population is in Hardy-Weinberg, right? So that's how you use it. And you might study that population for 20 years, I don't know, right? And you take samples every 20 years, and if the genotypic ratios don't change and the phenotypes don't change, then the population is in Hardy-Weinberg. Now, what you're going to find is that doesn't happen in nature, okay? Probably the population that's closest to that are humans, all right? So we can use Hardy-Weinberg to predict the probability of you having a baby with cystic fibrosis. 1 in 3,000 births, right? And that's just randomly, unless we know your family history. So, and that's because the human population is huge. We kind of control our environment. We assume there's not a lot of mutation, right? And we assume we randomly breed. And so we can get at least assumptions on a lot of genetic abnormalities that you see within our population and use those in genetic counseling. That's kind of how we use it. We use it as a null hypothesis. It gives us a mechanism to set up an experiment, right? That's what's important here. Then it would provide us data. And so let's say we take this population and we look at it the next time and it's 45 out of 70 white, right? And when we find that Q squared, that's going to be different than the original Q squared, right? Then the population's not in Hardy-Weinberg. Then you can go, well, what's going on? Then you can start to look closer, right? Does that make sense? So that's kind of how we would use it. We already did the hamster example, right? Showing you how we can count alleles. When you get to ecology with Dr. Sweets, you'll actually do some Hardy-Weinberg, right? He'll actually do some Hardy-Weinberg out in the ecological world. He loves Hardy-Weinberg, so as you'll see. So here's a good example. Here are wildflowers. So we have our wildflowers here. Notice this is incomplete dominance. So we have a homozygous dominant red, a homozygous recessive white, and then our heterozygotes pink. And if we knew the whole population, this is a great sample size, right? Out of a field, if you took 500 of these wildflowers, that's a big sample size. That's a nice one. That gives us 1,000 total alleles, right? Because remember, they're diploid. We have 2 copies of chromosomes, one from mom, one from dad. And if you added those up, and if we could do genetics and we knew their genotypes already, we could count the number of alleles and get our R allele frequency and our little r and big R frequencies, right? And we would do that using pretty simple math. Since we doubled everything, we have 640 of the big R's in our homozygous group. We have about 320, or we have 320 in our heterozygotes. That's 640 of the, it's 800 of the 1,000 total alleles are big R. All right, so our allele frequency for big R in this instance is 0.8, or that's P, isn't it? So that's P, which means Q is 20, right? Because we just subtracted from 1. So 0.2 and 0.8 are your allele frequencies for our population that we can then use if we want to keep studying this population. So if you wanted to predict, since we know this, what the next generation, in the next generation, how many heterozygotes we have, you could do that, right? You would just plug these 2 numbers into 2PQ, and we would predict that if we sampled next year, 32% of the dominant phenotype would be heterozygotes. And when we go and do that, if it is 32 or close to 32%, then it's in Hardy-Weinberg equilibrium. Okay? If it's different, then it's not. Does that make sense? So, as I mentioned, this doesn't happen in nature, and there's a reason for it. Because these are the assumptions that have to happen in order for any population to be in Hardy-Weinberg or not changing as far as its genotypes and its allele frequencies. We're going to assume if a population is in Hardy-Weinberg equilibrium, there's no mutation, there's no migration, there's no new pollen coming into our population of wildflowers. There are no bees carrying new pollen in. The population's very large. We have random mating, and then there's no evolution going on. There's no natural selection going on. There's no natural population that does that. Typically, natural populations have multiple of these happening at once. So we know this doesn't exist in nature, but it does work pretty well for us, for humans, to do some, genetic counseling and kind of get percentages of disorders that you see. So it's a nice mathematical model to test whether a population is evolving or not. So this is what it should look like if a population does not evolve. There's the starting population. There's my first sample. And butterflies are like flowers. In the winter, they go to, they bred and they die, and then the babies overwinter. Then this is the next summer. This is the third summer, right? And if the numbers remain consistent or close to consistent, then the population's in Hardy-Weinberg. So these are the two equations you will use on that third exam to answer questions about populations and their allele and genotype frequencies. So in the example I gave you, I kind of worded it a different way, right? So here again we have our flowers. If our population is 60% big R, right? We know that from our sample. Then we know little r, or Q, is 40%, right? Or 0.6 and 0.4. Those are our allele frequencies. And so if we wanted to know next summer what is our predicted heterozygote number for our dominant trait, we could just plug those numbers in, right? Now, if we go back here, in this example here, notice, if we want to know what our heterozygote prediction is for our next generation, then we want to use this part of our equation, 2pq. To do that, we have to know p and q, right? And if we are told this, that a population has 30% dominant plants, that means 36% dominant plants. That means 36% of the plants are the dominant phenotype color, right? What is the heterozygote percentage? To do that, we couldn't use this number, right? Because we don't know if dominant plants are homozygous dominant or heterozygous. But if 36% of all of our plant population is dominant, then we could subtract that from 1 and get the recessive number, correct? So, which is 64%. That equals 1. But 64% is the genotypic ratio. That's Q squared, isn't it? So 0.64 is Q squared. So we would have to square root that, which is 0.8. And then that would give us Q, and subtract that from 1 to get P. Now we're ready to go. Then we can plug those two numbers, 0.8 and 0.2, into 2PQ and say, next year when I sample the flowers, 32% of them should be heterozygotes, right? And if they're not, then we're not in Hardy-Weinberg. So the big thing is when you're doing these problems later, and I give you word problems, right? It's just like I tell my kids today. I tell my middle schooler, you gotta draw yourself a picture. Write out everything you know, right? Your big letter is red. Your little letter is white, right? Write out everything so that you can see it in front of you, and then you gotta use what you have, okay, to get your P and Q numbers so that you can plug them into Hardy-Weinberg, okay? Now, let's talk about the violations, because obviously in nature this doesn't happen, right? So there are minor and major violations of Hardy-Weinberg that throw our numbers out of whack, right? That give us different genotypic and allele frequencies from generation to generation. The two minor violations are non-random mating and mutation. Now, a lot of organisms don't randomly mate. They are selective. They select their mate. They're very selective on which mate they will mate with. They throw out some genotypes that determine certain phenotypes that they don't find attractive and breed with very select numbers of phenotypes that are controlled by genes. This will change the genotypic frequency really quickly if it's a strong selection pressure. Mutation can also change our genotype and allele frequencies, but it's much more rare. Remember, I already told you it doesn't happen very often, particularly in a eukaryote, because we have really good proofreading. Now, if we're talking about an RNA virus or COVID-19, then mutation is a major contributor, right, to the evolution of a virus or bacteria. But in animals and plants or eukaryotes, it's only a minor violator. Might take time to actually see it change your genotypic and allele frequencies. Major violators are these three: small population size or bad sample size. The smaller the sample size, the more common your Hardy-Weinberg equilibrium is disturbed. This allows for things that we like called genetic drift. So if you have, and I'll show you some pictures, genetic drift, there's a couple different types of genetic drift. There's what's called bottleneck and founder's effect. Those are your two types of genetic drift we're going to do. And we'll talk about how small sample size can affect what alleles you see here in a slide or two. Migration brings genes into your population. All right, we call it gene flow. Migration happens a lot in populations because remember, typically, got to remember a definition of a population. It's a group of a single species that is interacting and interbreeding, and a population that we consider a true species doesn't have to be one population. There's usually several of them, right, that are separated by barriers that are not true barriers. They can actually migrate between populations. Mm-hmm.
I said not just talk about interbreeding. Like when I think of interbreeding, I think of like incest.
No, I'm not talking about incest. I'm just talking about breeding within the population. Okay. Yeah. You don't want it. That would be severe selective breeding there, where you're breeding just within a family line, which does happen, right? I mean, it happens in humans all the time, and then their genes are really messed up typically, if it's many generations. That's why hemophilia is common in royal lines from Europe, because of inbreeding. What? Yeah, like in Habsburgs, yeah. Yep. It's like they bleed. They have bleeding disorders. Now, genetic drift, small populations. This is a big problem nowadays because we are fragmenting populations into small populations. And those small populations are separated by human development, for example, right? And so they're stuck somewhere and they can't move, so they have to interbreed. And when you take a big population of a bunch of alleles and you fragment it into smaller subsets, you're going to remove alleles from each population, right? Because you're going to separate them into these small populations. And if they can't migrate, that can cause genetic drift. And if you let those little populations breed for a couple generations, their genetics changes, right? And it would be an example of evolution driven by small sample size. Now, what we're trying to do as scientists nowadays is when we fragment a population, particularly if it's because of human development, houses, fields, roads, those types of things, we try to build corridors to allow the populations to interact, right? So for example, you have a big population and you put a 4-lane highway. that cuts the two populations in half. Now you have a population over here and a population over here, and they're turtles or something, and as they try to cross the road, they get run over. I mean, they try to migrate, but they can't. So what you can do is dig a tunnel under the road or a bridge over it that allows the animals to move, right? So that they're undisturbed, move between populations. And so that's what, they're trying to do that, for example, in Costa Rica, where I go a lot, where they cut down all of the forest and now they're regrowing it. And so they got, forest on that table and forest on this table, and then a big human development between them. And so they're building a forest corridor to connect the two, right? They plant trees. So they buy farmland and build a forest corridor, if you will, a forest road that allows organisms from one population to move to the other one, right? So you can get good genes coming in and out. Because migration's good. Migration brings good genes into a population that might be missing. So if you look at cheetahs or a lot of leopards in Africa that have been fragmented into small populations, their genome is really different than the original genome, and all of the animals are related, right? It's not a great thing. Their genetic variability is not very good. So what happens when you separate a population into small ones and you let that small population breed is your heterozygosity disappears, and you end up fixing the genes at loci into homozygous recessive or dominant. And it looks something like this. And they've done lots of experiments to show this. And heck, they've done wild experiments to show this. I mean, all you got to do is look at Africa. We've separated lots of big populations like elephants and cheetahs and leopards and lions into smaller fragmented populations. And you can just look at their genetics. But you can do this with rodents too in the lab. If you have a sample of rodents with good gene variability at about 1,000, notice the frequency of the big B allele here stays about the 50% range, right? It goes up and down, but it stays in the middle. If you take 10 animals out of that 1,000 and just let them breed by themselves, notice what happens, right? It goes all over the place and eventually fixes either this way or that way if you make the population really small. And that's because it's just like flipping a coin. If you take a coin and flip heads and you do it 10 times, there's a better chance of you flipping 6, 7 heads with 10 flips versus 100 and doing 50 of those as heads, right? Or 1,000 and 500 of those flipped as, or 600 or 700, 800, right? The bigger the population, the less likely that you're going to randomly cause the selection of a particular allele. So this is called fixation. When you fix a population so that all of them are homozygous dominant or all of them are homozygous recessive. You reduce the heterozygote. So how does genetic drift happen? First example is bottleneck. Bottleneck is when a huge population undergoes a devastating disaster and you remove 90% of the organisms. So you kill 90% of the animals, just like we were doing with the bighorn sheep, right? But we kill lots of them. We only end up with 10%. That means you remove 90% of the alleles that are there. The leftover population obviously doesn't have all the genetics that the original population has. In this example, notice we lost all the yellow beetles for whatever a rock fell down and crushed all the yellow ones. I don't know. So you only get the red ones, and that's all you see after that in the population. There's examples of this that happen all the time. Great example here in the U.S. caused by us, the American bison, right? Buffalo. In the 1800s, we killed hundreds of thousands of those, down to about 100, 200, 100 or 200 in a herd that used to be 100,000 animals. And then they became protected, and we let them breed again, and now they're 40, 50,000. If you look at their genetics, they're pretty related, right? I mean, they all came from that group of 10. Obviously, that group of 10 doesn't have half the genetics that the 100,000 had, right, originally. And so that's an example of a bottleneck. This monkey here, it's called the golden snubbed monkey. It's got golden fur. It's pretty. It's probably one of the most unique primate you'll ever see. It's got a blue face, no nose, so it looks like a blue skull. Its face does. Crazy looking. It's from North Vietnam. Think about that. North Vietnam, the Vietnamese War. It was pretty much extinct. All its habitat was destroyed during that war. But it has bottlenecked several times during its life history, and there are only about 200 of these left. So it's one of the most endangered primates in the world. And obviously, it can only breed with those 200 individuals, right? So it's a tremendous bottleneck. So why do they call it a bottleneck? So think of it like this. If you have a big container that has a limited neck, right? And you dump this over and collect 5 or 6 beads out of it, and you have all these different colored beads, obviously the 5 or 6 you get out is not representation, doesn't represent, I should say, the entire population that's in that bottle, right? These are the only ones that can breed now, and you're going to lose all of those alleles that you had. So that's considered a bottleneck effect. Happens a lot across the history of the Earth. We're obviously helping it a lot now with our destruction of habitat. Another way this can happen, which happens pretty naturally, is what's called the founder effect. It's when you have this huge population and a few of them decide to leave and form a new colony, right? So this happens a lot. You get a big population somewhere and it gets really, really crowded, and then a few of the more adventurous, in that population leave, and they go try to, form a new colony. And if that colony's far enough away and you don't get a lot of migration from the original larger population, then you might, get only a certain number of alleles in that small representative sample. And if they are able to interbreed for many generations, their genotypic allele frequencies are going to be much different than the original population, right? Great example of this happened during, there are several of them, there are bunches of them actually, happened during the expansion of the European population to Africa, South America, and so forth. One great one is a Dutch population that is pretty much Dutch and some English, that settled in a remote beach in Africa, South Africa. It's called the Afrikaner population. They have a really high incidence of Huntington's disease, which is a neurological degradation of the nerves that typically shortens lifespan once the disease shows up. It is a dominant trait. And I think anywhere from 50 to 150 people actually started this colony here in South Africa, and several of them were carrying that disease. Right? And so you went from millions of people and maybe just a couple of Huntington's diseases in Europe to 2 or 3 of them in 100 having Huntington's disease. They obviously passed that gene on, right? And it is pretty prevalent in that group, even though there is migration and whatnot that comes into that particular population now. So newly founded populations don't always represent the genetic diversity of the original population. Okay? That's called founder's effect. So those are 2 examples of genetic drift. Okay, as I mentioned, gene flow is a good thing because it brings new alleles into a population, or it will bring alleles that have disappeared into a population back if that population, which is separated from the original one, is under different selection pressures. And so it homogenizes populations if you have interbreeding between all of the populations. So gene flow is good because it keeps alleles consistent between populations. Migration or gene flow can happen with individuals or gametes. Gametes can travel in many cases, not individuals. Think of plants. Pollen, the male gamete, it's really the male gametophyte, can fly. It can travel. It can travel hundreds of miles in the air. It can be carried by bees hundreds of miles, right? And so, it's a good way to transfer your genes in a wide radius over hundreds of miles. So it reduces differences between populations and keeps genetic variability high. It also is going to prevent reproductive isolation and maybe prevent speciation over time, right? With groups, if you're looking at anything that's above microevolution.
So does that like increase genetic diversity?
It can, for sure, yeah. The last one here that is a major, it's the largest, and the major change that it causes, or the major mechanism that causes changes in gene and allele frequencies is natural selection. All right. The really nice thing about natural selection is it is non-random and directional. It's the only one of these that really is non-random and directional. A rock falling on 20 different beetle phenotypes and killing all of them except for 4 is pretty random, right? An asteroid hitting a volcano, it's random. This is not random. Natural selection, remember, nature's selecting for a phenotype, and that phenotype becomes common while the rest of them become uncommon, right? So it's directional for a particular phenotype. And it can happen slowly or it can happen really quickly. It just depends on the environment and what constraints are on that population at that time. Birds are great examples of this. These are honey creepers, all right? So we use Darwin's finches. These are Hawaiian honey creepers. So you can see that you can tell they're kind of all honey creepers except maybe the middle guy. But they all have different beaks, and those beaks allow them to take advantage of different food sources, and which allows them to not compete against each other, which is important if you want to survive. These animals then develop different colors, different breeding habits. The females use sexual selection to select the right male, and so that just drives evolution even faster, right? Or causing reproductive isolation. Pre-zygotic barrier, right? Like we talked about earlier. So how does selection happen? If we know it's the fastest, it's the most directional. It's also the major one of the that cause us to leave Hardy-Weinberg in the dust. Selection happens because of selection agents, or what we call agents of selection. I call them in selection pressures. That's the word I like to use. I don't like agents of selection. It's too many words. I just like selection pressures. But we call them AOSs, agents of selection. These eliminate non-adaptive organisms by causing death, all right, to other genotypes and phenotypes. It increases the frequency of adapted organisms over other phenotypes. There are a number of different agents of selection, all right, and there are basically 2 major genera, if you will, of agents of selection that cause natural selection. They are environmental or non-living agents of selection, and then living agents of selection. So here are the non-living environmental agents of selection. They are temperature, weather, water and salinity, frequency of disaster, and amounts of shelter that are provided in any one environment. Probably the 2 most influential across the history of, say, vertebrates, for example, which I study, are temperature and water. These 2 really can slow the ability of organisms to occupy an environment. So if you can deal with cold temperature and occupy somewhere that other animals can't, then you can breed and survive. Water's another one. Think about this. If you could drink salt water, I mean, how adaptive is that? Right? The planet is 75% salt water. If you can drink it, that's a big thing. We can't drink it. If you try to drink salt water, you'll die. Don't do it. Might as well drink your urine. You can drink urine, well, maybe 3 or 4 times if you're trapped before you become too dehydrated. So salinity and water, huge. Osmotic barriers are huge to the control of which animals can survive in certain environments. Now, once you're there, once you can deal with water, the second thing that you have to deal with as an organism are living agents of selection. And there are 3 types of living agents of selection. The first one is competition. 2 animals land and can survive in an environment based on non-living conditions like temperature and water, and then they find each other competing for the same food source. The one that is more adaptive and getting to that food source before the other is the one that's not going to starve, right? And so these are antagonistic relationships between 2 or more organisms over resources. Competition is huge and fierce within environmental niches. As a matter of fact, you're going to come to learn in ecology that one organism can occupy a niche at a time. Because if 2 are competing over the same niche, both pretty much do really poorly, right? So there are very few examples of organisms occupying the same exact specific niche. And that's why evolution drives who can occupy a niche based on phenotypes and who has the best adaptation. So these are some examples of organisms competing, right? Not eating each other, but competing over a food source. The next one is probably the one that's most familiar to you, which is being eaten, right? Or being the predator. So predators, prey, and you can add another P here, parasitism, right? Those are, I call them the 3 Ps. So these interactions can be positive or negative, depends, but they're relationships between 2 organisms where one preys on another one or one parasitizes another one. And those can be obviously beneficial to the predator and not beneficial to if you're getting eaten, right? The third one is called sexual selection, which is huge in a lot of organisms. This is where there's either competition between the males or females in a species, or one of the sexes is selective over the other. And so this is going to force the opposite sex, for example, to take on a particular trait or phenotype that the female, for example, finds attractive, right? Whether it's feathers, dance, call, like the peacock feathers, whatever it is that causes the ability to reproduce more successfully means you're going to pass your genes on. So we're talking about agents of selection, right? We did non-living, and then we started the living environmental agents of selection. And we have 3 of those. The first one is competition, right? So you have a community. A community is made up of populations of individual species, and those species obviously interact, right, within that community. And that interaction can lead to selection events. So what are called agents of selection, living agents of selection. The first one is competition if you're competing for food, right? We talked about that one. The second one is the 3 Ps, remember? Predator, prey, and parasite. And then the third is sexual selection, where we have mate choice. There are 2 types of sexual selection. There's intraspecific, which is between the same sex, like male-to-male combat or male-to-male competition. The inter part here of sexual selection is between the sexes, male and female, and often has to do with mate choice, where the sex is choosy in selecting who they will reproduce with. And I gave you some videos here you can watch on males trying to perform and grab the attention of females. Happens a lot in birds, a whole bunch in birds, and even in invertebrates like spiders. So here's a peacock. Male peacock has the larger the plumage, the more sexy he looks to the females, right? So the reason females do this is because of what's called the good gene hypothesis, which I think we mentioned right at the end last time. And this has evolved because females have this huge disparity in the amount of energy that they use to make eggs and also care for young in many cases. And so if they're only going to breed once, let's say a season or maybe twice, and they're producing eggs, then they want to be really choosy because remember, males have the choice to breed with whoever they want. And they can spread their genes around, right, to females, theoretically. Where females, hey, I got to breed with one male, fertilizes my eggs, that's all I got, right, maybe in a season. So we want to make sure we pick a male that has good genes. And males that can flaunt, dance, and show off this beautiful plumage while surviving everyday life, escaping predation, and still gathering food and having enough energy to breed must be a good male, right? And this leads to distinct sexual dimorphism, which means the males are typically brightly colored, beautifully plumaged, where the females look more cryptic and drab and easier for her to hide. And then the male has to go through this ritual, these behavioral rituals, these dances in order to gain female attention. And as I mentioned, this happens because of this asymmetrical parental investment, where not only is the female producing these eggs, which takes a lot of energy, but she also spends time in offspring care in many cases, particularly with birds. So females incubate in many cases. The males help sometimes, but the female really not only makes the eggs, but also incubates and then cares for the young for a particular amount of time. Because of that, she's limited on her mating choices. And when you add all this up here, cost of egg, cost of pregnancy, parental care, it's much greater cost than, say, producing sperm and breeding with multiple females. Here's a great example of this. So this is a kiwi bird. It's a bird that's about this tall. It's about that tall. I visited New Zealand in the late 1990s and got to see a kiwi. So there were a couple kiwis out in the wild. It was pretty cool. They are a flightless bird. They use their long beaks to go through the sandy soil of its habitat, removing invertebrates for food. But the females produce an egg that's about as big as their body cavity. And it's one egg per season. And so they got to be really choosy on the male that they decide to breed with. So now that we know that selection happens, and we got these agents of selection, whether they're environmental or they're biological or both, they lead to changes in phenotype frequency, right? Remember, selection works on phenotypes. Phenotypes are driven by the genotype you have, correct? It's kind of where we've talked about to this point. There are 3 shifts that you see in microevolution. If you're following a population, there are 3 kind of shifts that you can see in phenotypic frequencies, and these shifts are non-random. They move directionally. And if we use this kind of example here with these modes that we see, if this is our original population, and notice we have a bell curve here, so this is a phenotype or phenotypes that are controlled by multiple genes, right? This is a multiple gene, or what we call continuous gene setup, where we have several genes that are giving us little small increments and differences in coat color and maybe coat pattern here in rodents. And we obviously start off with an original population that looks like this. And selection is going to happen to this. And the selection agent in this instance is a predator. Okay, so notice the hawk here eating the mouse. So there are 3 ways or 3 shifts that you can see. The first one's called directional selection, where we move either to the right or to the left. And the phenotype is going to shift one way or the other, depending on the environmental selection agent or the biological selection agent. With rodents, it's pretty simple. It's all about camouflage, right? And so you can see over here on your left, you have a light substrate, and on your right, you have a dark substrate, right? And obviously, this guy hides better on the light substrate, but stands out on the dark substrate. So if you have these 2 environments, 2 populations in 2 different environments, one population is in a dark substrate, one population is in a light substrate, you can imagine you're going to move directionally over generations based on color, right? Those that can hide the best on the substrate are the ones that are going to survive and reproduce. Pass those genes on to their babies, that also survive, right, if they're dark colored. And you get this directional shift to the dark phenotype on the dark substrate and the light phenotype on the light substrate. That's called directional selection. The next one's called disruptive selection, where you select against the middle ground and push your phenotypes to the extremes. And you typically see two extreme phenotypes. So in this instance, it might be some selection that selects for the lighter coat and the darker coat, but not the middle coat color, right? We see this in nature also, where you get these phenotype extremes and selection against the middle phenotype. It happens a lot in birds, particularly birds on islands that eat seeds. Often what you'll see is you'll have a bird with a small beak and a bird with a very large beak, but very few birds in the middle with kind of a middle-sized beak. And this is because the smaller beaked individuals can manipulate small seeds and crack them and eat them, like something a sunflower seed size. And the larger beaked individuals can't handle those small ones, but can crack the bigger seeds, eat the harder, larger fruit. But typically the middle ground beak can't do either, right? So it's selected against in this instance. And this is called disruptive because you're disrupting or moving away from the middle of your bell curve here. Number 3 is called stabilizing, where you select for the middle ground. Yep.
Can you please slow down a little bit? I'm a little behind.
So, oh, you want me to slow down? Oh, well, okay. Okay, as I mentioned before, the third one here is what we call stabilizing selection. So in this case, we're going to choose for the middle ground group and select against the extremes. And so we're stabilizing the middle, or what we would term maybe the heterozygote in a single gene situation. In this instance, with the mice we've been talking about, we picked the middle ground mouse here, the middle color, the color that's between the really light and the really dark. We see this a lot in nature also, but one really good example with humans is birth weight. So when you look at birth weight across the human population, it forms a really tall curve here where the extremes are selected against. That is, babies that are born very light often die either right after birth or pretty recent after birth, and the same with really heavy babies. And so the babies in the 6 to 8 pound range are the ones that have the greatest survivalship. And so it's obviously the middle ground you're selecting for and the extremes you're selecting against. So those are the kinds of the 3 ways selection happens in nature when we're talking about moving around during microevolution. So we have directional, where we move either right or left. We have disruptive, where we move to the extremes, both extremes, or stabilizing, where we select for the middle phenotypes. All of these driven by agents of selection. So the question might be, well, how do we get new phenotypes, right? So phenotypes are driven by genotypes, which are driven by alleles, correct? So a lot of new phenotypes can come into a population because of mutation. One other way this might happen is maybe migration. Remember, with migration, some individuals from a different population come in and deliver some new genes, right? Deliver some new alleles. But in any case, when new alleles do arise from mutation or migration or some other way, then we're going to get new phenotypes. And new phenotypes can sometimes lead to new adaptations. So the big question is, do all phenotypes equal adaptation? And obviously we've already answered this question once, and that's no, right? That's not always the case. That is, every new phenotype doesn't lead to better reproductive success, right? And if you look at a coat color is a good example. I just wanted to show you that there are several genes on the chromosome, it's called the agouti chromosome here in rodents, that control coat color, pattern, dorsal striping, ventral coloring, tail striping, penetration of the color, how dark the pigment is, how bright the pigment is. And these genes, there are several of them, are spread out along this chromosome. And so you can see there are a couple dots here that represents a couple different areas or genes that are responsible for seeing these color differences. So here's the light phenotype on one side, and here's the dark phenotype on the other. And these colors, which are found down here along the chromosome, are the genes that control those phenotypes. All of those genes together give you that continuous variation of phenotypes that you see. I'm only showing the light and the dark here, your two extremes. So mutations in these could change color pattern, right? They could change color pattern. You can get a completely white mouse, for example, right? If all of these genes don't work, like an albino. Or you could, change how wide this ventral coloring is on the ventral surface versus how bright the dark is. There are a bunch of different ways a mutation could slightly alter what you see. Those new adaptations, if they help with reproductive success, are going to be new adaptations. Okay? So how do we measure that? We call it fitness, is basically what we call it. We call it relative fitness. And relative fitness simply means that if a new phenotype arises, the big question is, does it increase your relative fitness? And what is relative fitness? By definition, relative fitness is how many of your offspring survive in the next generation. Right? First, you have to produce them. And then, do those offspring survive? And how many of them then reproduce to produce another generation? And so forth. So relative fitness is first, do you reach reproductive age? And do you produce babies? And do those babies survive and produce new babies? Okay? And if phenotypes increase your relative fitness or your reproductive success, then yes, they're adaptations. If they don't, then they're just a new phenotype. Now, new phenotypes are maintained in a population because we like genetic variation, right? And therefore, phenotypic variation. The reason for that is because if a new selection pressure comes along and a hawk likes light mice, for whatever reason, it might eat all the light mice, right? Leave the dark mice alone. Then it's a different selection pressure. Now, a phenotype that wasn't advantageous is, right? So having that genetic variation and those phenotype variations within populations allow you to deal with new selection agents or new stresses. So that's what relative fitness is how successful are you reproductively. So I always joke at the end of this slide that I have good relative fitness so far. I have 4 kids. Dr. Milne, one of the other teachers who usually teaches this class, has no kids. So he has no relative fitness. I have decent relative fitness as long as my babies have babies. Not yet. They're all too young. But eventually. Dr. Milne has lots of cats, I think.